And now we're going to talk about sequence variability and how important the sequence actually being exactly right matters for function. And the answer is very little. So given that sequence is the code for structure, and structure needs to be maintained for proper function, it seems that there'd be no room for variability. The reality is that that is completely kind of wrong, that you can actually have a lot of variance in the sequence, and we'll look at that in a second, and still get function out. Okay? So sequences have a lot of freedom and variability, and this was first explored with something called the Globin fold from the Globin family of proteins. The Globin family all have 8 alpha helices, typically labeled A through H, and they have a roughly the same 3D arrangement of the helices. Okay? So here we have myoglobin. Here's the hemoglobin alpha chain, the hemoglobin beta chain, and then we have some hemoglobin derived from other species, so worm hemoglobin, clam hemoglobin. This one's from a plant, like a legume. And again, you can see like some slight differences. The length of some of these alpha helice barrels are a little bit different. Maybe their rough orientation looks a little bit different, but overall, they still have 8. They're kind of the same shape. They're all around a heme group. Again, the heme has the ability to bind oxygen. So typically, they look pretty similar. So sort of this, despite near perfect structural conservation, okay, so this overall structure looks very similar across the board. The underlying sequences are quite different except for a few key amino acids. Okay? So this is the top line. Oops. Is human hemoglobin beta chain. We have hemoglobin alpha chain, and we have myoglobin. The dots represent gaps in the sequences, so they're not exactly the same length, right? So the dots represent some sort of gap or missing spot. And then the green represents sequences that are identical residues. Okay? So what percent is identical, would you say, out of these 3, roughly? Just estimate the amount of green you're looking at out of all of them. I heard 15. Any other guesses? 20. Great. So sequence identity is the number of identical residues you have, like in the same position. These are all MMM. These are all leucine, 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 valine, valine, valine. Same position from 1 to however many there are. It's the exact same amino acid residue. Identical over total number of residues. So for these, we're looking at roughly 20 to 30% identity, which is pretty low, considering how similar they're able to fold and what functions they're still able to do. So here we are, all of the ones from that other figure. So now we have our top 3. We have our alpha, beta, and myoglobin chain. And then we have sort of our other species listed here. Okay? Same thing, dots are representing some sort of gap in the sequence because they are slightly different lengths. The colors are indicating decreasing similarity, with green being exact matches, and then as you go towards sort of like this, I don't know, ugly looking lime, and then you go into more like yellow. This is a weird way to do it, but they're representing through to white, which is like few or absolutely no matches in that particular position. Okay? And so what this is showing is that there are only 2 absolutely conserved positions across all of these. Okay? A very small number of amino acids were conserved across all the globins. Okay? The ones that were absolutely conserved, so they're exactly the same position, same amino acid across all of them, were only 2 spots. A phenylalanine in the 44th position and a histidine in the 88th position. Okay? Were 100% conserved. They were not varying, not invariant. I only phrase it this way because you might see it written this way on other questions, and so understand that I'm asking the same thing. Okay? So the reason these have to be in the positions they are and they have to be the residues they are is because they interact with the heme group. They're essential to the function of the hemoglobin or the globin molecule. Okay? And so they're sort of shown here. We have the phenylalanine and we have the histidine. Okay? They both interact with the heme group. Okay? So where they are located and their ability to interact with the heme group makes them essential. They have to be conserved. But everything else is kind of flexible.